Hey guys, join me for this ride. I'm going to talk about Alina Chan's recent paper that just got released. I don't get to cover all the figures because the bike ride is very difficult and it's windy and I lose my train of thought a couple times. The first figure is very simple. I cover that one in the ride. The first figure basically shows you a phylogenetic analysis of the amino acid changes that occurred in the early versus late SARS-CoV virus outbreak in 2003 versus the SARS-CoV2 virus, which is the current pandemic virus. What you notice there is despite the fact that SARS-CoV, the original one, didn't go through nearly as many victims, it showed a great deal of phylogenetic, or let's say amino acid sequence change during its brief course on Earth. In comparison, SARS-CoV2 has gone through many thousands more people, but shows far fewer changes from an amino acid to amino acid basis. That's basically figure one. Figure two is, let me think about it for a second. What is figure two? Figure two is the, where is figure two? Let me think about what figure two is. Figure two is, yes, this plot with all the dots on it. And that plot with all the dots on it is the plot that's trying to show you the sort of time course, the change, the relative rate of change. And the relative rate of change in early SARS was higher than the rate of change in late SARS. And that rate of change in SARS-CoV-2 is about the same as late SARS. That's what figure two is showing you. Figure three is then showing you the SARS spike protein and the changes that occurred during the SARS spike during the SARS outbreak, early on the bottom, late on top. And then the final figure four shows you the changes that are occurring in the spike protein of SARS-CoV-2. And again, far fewer changes are seen despite the fact that they have so many more sequences. And then figure five is showing you a cartoon diagram of how a typical pandemic is thought to go out and how that fits in with SARS versus the current SARS-CoV-2. If you look at the figure on the right and you see, sorry, on the left, and you see the original SARS, what you see is that we have several different pieces of the puzzle which collectively make sense as a sort of zoonotic explanation for how SARS emerged from nature. And for SARS-CoV-2, we have none of these pieces, even though many papers have come out in the last 3 months purporting to give you these pieces. Alina and her lab's analysis is showing you that none of these pieces fit into where the typical pieces of a zoonosis should fit. So she makes the argument again that we need to make a more concerted effort to find the location, the source of this virus. And she, very controversially for some, makes a distinction that means that we also have to consider the fact that a laboratory origin of this virus, because it's so well adapted for humans before we even noticed it, that the lab origin cannot be ruled out. So join me for the ride. The wrap-up's pretty good. Evil, and we need to get on Alina's side and make sure that Alina Chan knows that the American people are supportive of objective science and objective interpretation of your own scientific results. I don't think Peter should be on Twitter telling Alina to change words or change the way that she interprets things. And notice, if you take a very close look at Peter Daszak and what he is doing on Twitter on a daily basis, you will see a guy who gets paid $300,000 a year to run a nonprofit organization that really just accesses grant money from the United States government, from shady parts of the United States government, NIAID and USAID. You can look those up, especially the U.S. Agency for International Development, and see that there's no reason to believe that funding from that organization going to Peter Daszak and then going to this gain-of-function research in China should be looked at as anything benign. That needs to be investigated thoroughly. So anyway, if you liked it, like and subscribe. Enjoy the ride. I'll see you next time on JC on a Bike. Good evening, Pittsburgh and universe. This is JC on a Bike. My name is JC and this is Journal Club on a Bike. And this evening we're going to be covering a bioarchive preprint from Alina Chan's lab at the Broad Institute at MIT in Boston. This paper has caused a lot of controversy on the internet in its short period of time of being available, simply because many people see this as direct evidence for a possible alternative explanation to this SARS-CoV-2 outbreak. And the way that this paper approaches the question is to try and use the previous SARS outbreak as sort of a gauge by which to try and figure out at what stage of this zoonosis we're at. And to preview this, I think it's important to call attention to this paper by Baric's lab. I don't remember the year, but I'll flash it up here and you'll be able to read the paper, where he and the usual cast of characters took a Civet SARS virus, a Civet coronavirus similar to SARS, with a similar spike protein to SARS that was adapted for the Civet ACE2 receptor. And they adapted that spike protein sequence to infect cultures of cells that express the human ACE2 receptor. And what they found in that paper was that the changes that allowed the spike protein sequence to adapt, to activate, or to interact with the ACE2 receptor in the human cell culture, essentially those changes then ended up preventing the back infection of the Civet ACE2 receptor after these mutations took place. So in other words, this paper purported to demonstrate that zoonoses such as SARS are largely one-way streets. In the sense, that guy was signaling to go right, I thought, but whatever. In the sense that the structural changes that are required to move from one species to another most often preclude the back infection of the previous animal. So if it goes from civet to human, the change that's necessary in order to move from civet to human also makes it a one-way move. That you lose effective interaction with the ACE2 receptor in the civet when you gain it for the human. And I think it's an important thing to understand, because that's what this paper is trying to look at is let's look at what happened in 2003 with SARS, and let's look at the changes in the genome that occurred. And then let's look at the number of people that it went through, because that's another interesting, source of a discrepancy that I'm not even sure really Alina hints to very much. She does a little bit in terms of talking about figure 2, but SARS infected about 8,000 people, and it killed about, in the hundreds. But during that time, we have samples of early and late outbreak viral sequences, and they've been well taken care of. And so we can look at in figure 1, in part A, we can look at a tree that just describes the amino acid differences between sequences that were sampled during the outbreak of SARS in 2002 and 2003. And so that's what you see in the first panel in A. And that is compared to samples that were taken January, February, and March for SARS-CoV2. And now remember that SARS-CoV2 has infected many, many thousands more people in many, many different geographic locations relative to the SARS outbreak. So because the SARS-CoV2 virus has gone through so many more hosts, human hosts, than the SARS-CoV2 virus, evolutionarily speaking, one assumption or one expectation might be that if the virus SARS-CoV2 originated in the market in January or December, then we should be able to see the sort of stepwise zoonotic change that occurred from the beginning of the outbreak to now, where the SARS-CoV2 has this incredibly well adapted spike protein, can't tell what you're doing. This incredibly well adapted spike protein for the ACE2 receptor for humans, 10 to 20 times more affinity than the original SARS virus. And now, so what she's asking is if a zoonotic event occurred in the similar way as other zoonotic events, then there should be this transition period of different mutations which allow the gain of function in human and the loss of function in the previous animal, bat, civet, wherever it came from. That's how zoonosis works. And so there should be an evolutionary record somewhere, and there should be a mathematical relationship between early and late sequences and between viruses that are more closely related, then you should see less changes, this kind of thing. But through 800,000 to 1 million hosts, SARS-CoV2 has gone. So just from the perspective of viral genetics and viral evolution, if there was improvements to be made through those hosts, you would see stepwise changes, stepwise improvements. And as the selective pressures decrease, then you would see less changes. And so the first striking thing in figure 1 is that even though SARS-CoV2 has gone through many, many thousands more people, it shows a drastically less change. And you have to understand that how that happens is that in your body there are thousands and thousands and thousands of copies of the virus made, and not all of them are perfect, because the viral genome makes mistakes when it replicates, and those imperfections are selected against, right? If you make a mistake and the virus now sucks, that virus isn't going anywhere. If you make a mistake and the virus gets a little better, I'm glad I was going slow there, then that virus, that copy of that virus, that sort of variant of that virus could be selected for in an individual host and become the predominant variant in that host, so that host passes on this, gain of function variant of the virus to the next person that he or she infects. That's the idea. And if there's a lot of room for improvement, like there is after most zoonotic events, then early in an outbreak, you're going to see a high speed, rapid change, because any advantageous mutations that occur in the early hosts, in the early replication cycles, will be selected for, because they will by definition be able to infect more of that host cell and make more copies of themselves than the less efficient virus that first entered that host. And so, because zoonosis is imperfect, we would expect early in the outbreak to see much more changes than later in the outbreak, when the virus had settled on an effective, best sequence, and all the other sequences that should get made by accident suck in comparison. So then you see very little change. And that is what you saw in the late stages of the SARS outbreak, that those viruses were changing a lot less, and they were very effective at transferring from host to host. The pressures to change the sort of design space left had been greatly reduced, right? They're already doing pretty well. There's no reason to change. Most changes were going to be detrimental. But in the beginning, more changes can be fundamentally beneficial, because there's more room, because you're not that good. Your affinity is not that good when you first zoonotically jump. And so in this figure again, in B, they show you that the total number of changes relative to end-to-end comparisons. So you can see the differences in the early SARS viruses can be as many as 80 different amino acids. And in SARS-CoV2, the average is around 10. So there's so much less variation, despite the fact that there's so many more hosts. And we have so many more opportunities for evolutionary change. But there doesn't appear to be any real selective pressure on variants of SARS-CoV-2 in the sense of there's not much more to be gained. It's already optimized for human. That's the point. So figure 1 in the C panel, the one with all the funny shaped plots, that's showing you changes in different proteins that are synonymous versus non-synonymous mutations across only SARS-CoV-2 viral genomes, about 150 of them. So what you're seeing here again, and you want to look very carefully at the synonymous versus non-synonymous mutations. Remember, we explained that before with the Nerd Has Power blog, that synonymous mutations don't change the amino acid that is coded for, and non-synonymous mutations do change the amino acid that is coded for. And so in these proteins, if you look for non-synonymous versus synonymous mutations, the non-synonymous mutations are the ones that are be more interesting, and if they get selected for, then that would signify change. Whereas synonymous mutations, those are changes that don't matter for the progression of the virus. Now also draw your attention very clearly to the y-axis of these figures, because in the y-axis the numbers are not all the same. They try to show you similar banjo plots, or whatever those things are called, violin plots. They try to show you similar distributions there by changing the scales so that you can compare them, but the changing of the scale makes it actually very difficult for you to see how many more synonymous mutations there are. Especially look at the spike and the spike receptor binding domain, the first 2 columns. You'll see that the number of non-synonymous mutations is extremely low. And so the spike is barely changing at all. And that's not really how it should work unless it's already optimized. And being already optimized is a very good sign that we do not have sequences from the beginning of the outbreak. Let me say that again. The fact that there isn't very much variation from the beginning to the end, if you compare the first sequences from Wuhan to the sequences now in America, there's not enough variation to suggest that the zoonosis happened around that first sequence. Because if that was the case, then we would have seen the evolution, right? The change from. So the first sequence in the sequence of sequences is too similar to the late versions to make sense from the perspective of zoonosis. We should see a sort of reverse exponential decrease in the number of changes that are selected for as the virus adapts its previous host to its new host. And if you don't see that, then evolutionarily speaking, you are already far away from the zoonotic event. So all of the sequences that are in Wuhan are far away from the zoonotic event. That's the argument. They're not close because the virus was already done changing by the time we started sequencing it. Do you see? And so this presents a problem for all the market people, because the market origin theory has a very distinct timeline. The market origin theory can't be pushed back to October, because if the market origin theory gets pushed back to October, then now we have months and months of time where China wasn't warning anybody and China wasn't being open and clear about the dangers. And in December and November was when the Chinese government was investigating the Wuhan labs for leaks. In October was when all the cell phone data from the Wuhan lab went silent. So the moment we start pushing the outbreak back before December, the market theory falls apart, because as Alina points out, all the market samples just as bad. There's nothing new there. It's a human virus in the market already. It's not a zoonotic virus in the market. And so that's a big problem, because remember that there's been a lot of analysis on line 2 of this latest paper, which claimed that they found a bat virus that had an interesting spike protein with partial, furin cleavage site or something like that. And sequence analysis and lining up of various bat viruses by, let's say, non-biased molecular biologists has shown that the best alignment is not the one that they propose, because remember, all the bat viruses don't have one of these cleavage sites. So what they did, it looks like, there he is, that he, that's hilarious, that he, that was aligned then, right? So they just scooted a few of these amino acids over like beads on an abacus, and now, if you don't look carefully, you won't say, oh wait, those amino acids belong over here. You'll just take their word for it that those 3 are deletions and these 3 are right. It's not a continuous sequence, and it's extremely far from definitive evidence, especially when we keep borne in mind that these keyboard sequences, or that sequences can be generated on a keyboard. You have to keep that in mind. Now, Peter's never going to say that. Peter says they never have virus in the lab. Peter says there are no isolates. But Peter is a funder. Peter's entire existence is helping Chinese people get grants from NIAID and from USAID. And USAID is somebody that you better look up. So I'm not going to sit here and spout a bunch of conspiracy theories. But the United States Agency for International Development is far from an innocent organization in our history. There's lots of shady business in the history of USAID. And National Institute of Allergy and Infectious Disease is captured by Big Pharma. We know that too. So who's really funding this research through Peter Daszak? The guy who writes the grants, the guy who writes the manuscripts. It's a super shady nonprofit organization that pays their head $300,000 a year. He gets paid more than my head of department. In my head of department, Peter Strick, has a H factor of 88. Like, he's got a neuroscientist level, physics level H factor. He's got many articles that have been cited thousands and thousands and thousands of times. He's one of the foremost experts in monkey muscle physiology, and he's now working on the connection between the gut and the brain. That guy doesn't get paid $300,000 a year, and he gets NIH money, and he distributes it to labs in America at the University of Pittsburgh. He helps write grants. But he also comes up with the ideas, and he advises his students and his postdocs, and he critiques their ideas and helps them form the ideas that they'll use to go forward in science. Listen carefully. Peter Daszak does none of that. Peter Daszak does none of that. So thanks for joining me on this journal club. In this journal club, I'm covering the Alina Chan paper out of the Broad Institute at MIT, which has done an evolutionary analysis of the original SARS-CoV and compared it to a similar evolutionary analysis of sequences taken from the SARS-CoV2 pandemic virus. This paper has gained a lot of traction on the internet and caused her a lot of headache on the internet. And so the first call to action is come to Alina's rescue. Go on Twitter, post your support for her work, post your support for her gutsy, preprint and putting it out there, probably not realizing how much of the hornet's nest she would stir up, but now she knows for sure. And she needs our support to continue to do the objective research that she's doing, to interpret her results in the objective way that she's doing, and to not change the text of her paper because some loudmouth like Peter Daszak attacks her on Twitter and tries to publicly out her as a poor scientist when he himself is not even a scientist but just a grant writer. Peter Daszak is one of the nastiest people in this story, and the sooner that he is out of our ears and out of our minds, the better. Join me for this ride where I'm going to discuss this paper, look at the figures, try and get through it. I actually don't make it all the way through the paper because the first figure is really the money figure. The last 5 paper figures or so are very interesting though. 3 and 4 compare the changes in the spike protein of SARS-CoV and SARS-CoV2 and look at the differences that happen early and late in SARS-CoV versus what's really not happening early versus late in SARS-CoV2. This is all despite the fact that there are over 800,000 people infected with SARS-CoV2, whereas only 8,000 people were infected with SARS-CoV. That means despite not having very many hosts in which to mutate, SARS-CoV showed a great deal of change during the early versus late epidemic that SARS-CoV2 has not shown us. Alina Chan's lab then makes the interpretation of this data to suggest that the viruses that we have so far been sequencing are not viruses from the beginning of the zoonosis, but instead are already significantly after the zoonosis when the virus has started to stabilize. This means that we need to push back the date of the zoonosis, which means we now go into the shady region where, number one, the Chinese government was investigating the Wuhan lab for possible leaks. 2, we have this international event in October where military games where many different athletes from many different countries are purported to have been sick in December. And then we also have this idea that Event 201 took place in October where all of the websites, including the Next Strain website and the Johns Hopkins viral website, got rolled out in perfect timing for this pandemic to start in January. So we're really at a crossroads here where we need to continue to demand, continue to put pressure on the people that are being amplified right now as a means of sort of tampering down the doubt that this is a zoonotic event. They want us to believe that we need saving. We need to be saved from Mother Nature's evil viruses. And we need to find out once and for all. There's an Australian group that is starting to try and call for an independent investigation into the origin of this virus so that we can put our scientific and medical communities on the right footing. If this is a laboratory engineered virus, it would explain so many of the aspects that are currently right now leaving scientists with blanks. If you liked it, like and subscribe. Enjoy the ride. I didn't cover all the figures, but I'll try to do that maybe in a following ride when I wrap this up and continue it in the morning. Thanks again. See you soon.